Our first presentation today is Developing Integrated Pest Management Field Guides for Small-Scale Berries. Dru Yates and Marjo Dessureault are both joining us today from ES Cropconsult to share their work and answer your questions. And Dru is a consultant with ES Cropconsult, where she works on farms throughout the Fraser Valley in pest and nutrient management. And Marjo Dessureault is an IPM specialist and research director with ES Cropconsult. Marjo is located in Pemberton, where she provides IPM services to local farms. So as I mentioned, after Dru's presentation, we will have a quick Q&A session before we move on to the next presentation. And with that, please join us, Dru and Marjo, today. And Dru, I'll hand it over to you.
Great, thanks so much, Nadia. So yes, I am here to present about this project that both Marjo and I have been working on. Marjo is actually the lead, so she's here to chip in at the end during the question and discussion segment of things. So thanks, Marjo, for hanging out on the call while I give the presentation. So I'm here to talk about this story of us developing IPM resources for small-scale growers focused in berry production. And so this story is about 12 different field guides that we developed in the project, 6 of which were for berry crops, and those are set to be released in 2021. And part of what I want to talk about is that we did it in a bit of a novel way. So we spent 2 years, 2 different seasons working directly on farm with growers. And yeah, we had this focus on small scale operations. So yeah, I'd like to talk about what worked well and also what we learned along the way in this process. And before we get started, I have a question for the group. The question is, who here knows this poor sucker that I got to take a propaganda photo for our project? No, the actual question is, thanks for bringing it up, Shauna. Have you ever participated in making a fact sheet or a field guide or some other management resource for growers, either because you were making that field guide yourself or because you were a collaborator in some way? So I'll just give everyone a moment to respond to that. All right, what do we got? Ooh, an interesting split. Okay, this is excellent. So I'll be talking specifically about the experience that we had in this project, but part of why I wanted to ask that question is because I did suspect, and it does seem to be true, that there are some folks out there in the audience who have some direct experience with this type of work. And so I'm really curious to hear about people's experiences. And I really want to know how our process kind of compares to yours. So when we get to the question period at the end, I really look forward to hearing people kind of speak to that. Okay, so for these field guides that we developed, there's been this 3-year process. In 2019, we're really focused on the creation of some drafts of these field guides. This last year in 2020, we spent our time really doing test runs of those field guides and trying to tweak them to make them better. And looking forward into this next year, we are going to be releasing them out into the world to do their thing. So that is the general timeline. And as I mentioned, we had these farm visits as an important component of this project. And so we had farm visits that spanned throughout 2019 and 2020. So before we get too far into talking about the field guides, I want to talk about small scale and what do I mean by that. So small scale obviously can mean a lot of different things. And the way that we approached it in this project, we defined small scale as being less than 15 acres in total production. However, really what we were going for in terms of the small scale audience that we were trying to serve is operations that are sort of smaller than typical, what would be considered typical for that industry, or what is sort of typically the target for a lot of management resources that get developed. Which brings me into, why we chose small scale. And it does have to do with small scale often being sort of this underserved area when it comes to particularly IPM management resources that are available. We've identified that there's a large gap in pest management activities for small scale farms, all the way from limited pest monitoring to a lack of available resources. And obviously, small scale operations are highly variable, and that can make it really hard to target resources to actually be useful, not being too general and too broad, but also being specific enough that it can be useful. So that's what I mean when I'm talking about small scale here. And what am I talking about with these field guides? So before I tell you the story of how they got made, I do want to show you what a field guide looks like. So I'm going to pull up a draft that we are just working on finalizing right now. So these field guides, I just want to point out at the top, as I say, we've really targeted these for small scale operations, but truly they are IPM guidelines, integrated pest management guidelines that can be geared towards all types of productions. The principles kind of apply across the board, but we did really work to make this inclusive of and accessible for small scale operations in particular ways. So in general, we've got often at the top, there's a section about life cycle and general identification. On some of these field guides, there's identification that extends beyond the pest directly. So there's some damage identification for this particular field guide for two-spotted spider mites. Predator identification for beneficial insects in the field was also an important one for this particular pest. So sometimes the identification extends beyond just for the pest itself. The next section is often a section about monitoring. And this is usually where we really kind of tailored things more for a smaller scale audience, try to provide monitoring information that was applicable for small scale. Then we have another section that's really based around decision making. So trying to decide when you need to implement management and what factors you need to take into account to make that decision. And then the last section in these field guides is about management. So options around how to manage that particular pest. So that's a general layout of what I mean when I'm talking about these field guides. And what we're really going for is compiling information into one place so that it could be easily accessed. We're not necessarily discovering new methods or discovering new ways of management. We're trying to bring existing information together in an accessible and useful way. And another part of this project that's pretty important is this approach that we use, which was a really strong sort of emphasis on give and take. So we had these farm visits over 2 separate growing seasons where we would come to the farm and we would spend time asking the growers questions. We're asking them for some of their time and expertise. And then we would spend time after that directly monitoring and providing integrated pest management services to those growers. So giving them a bit of our expertise in exchange. So we were valuing growers' time in a different way than tends to be the case in projects like this. And this allowed us to gather information in a lot more of a direct way. It was very valuable on both ends. Okay, so how did we do this work? This is the list of growers that we collaborated with over the 2 seasons. We did this project in both the Fraser Valley and the Pemberton Valley. Marjo is based out of Pemberton, so we have representation in both of those places. And really the way that we chose to work with growers had to do with basically growers that would implement IPM principles if they were more accessible to them in some way. And so that was sort of how we prioritized some of the people that we would collaborate with. And one thing I want to point out in this list is that, so we have 3 different berries that we were trying to represent. We have blueberries, strawberries, and raspberries. And you can see that there's a straight shot all the way down for strawberries. And that's because we did this project for vegetable growers and berry growers. And for most of the mixed vegetable operations that we were working with, if they had a berry it was strawberries. So there's a lot of strawberry representation in this project as a result. Okay, and so the first thing that we did in 2019 in having these conversations with growers was trying to gather information about which pests we should prioritize, which we would actually include in our project to make field guides for. And we made this decision based on four main components. Firstly, our observations in the field, either our observations or the observations of the growers that we heard from them, trying to understand what were the problems they were facing. We had some consultation with the BC Ministry of Agriculture. A big thank you to Carolyn Teasdale, the berry specialist who really provided a lot of insight to us. We also then thought about which resources might be lacking for a particular pest. So for those of you who are familiar with pests in berries, maybe glaring omission to you on this list is spotted wing Drosophila, or SWD. And the reason that we did not choose this pest for this project, even though it is arguably the biggest challenge to IPM in berry management that exists these days, we didn't include it because there's a lot of energy, there's a lot of effort, there are a lot of resources that are being developed specifically around this pest. So we decided not to prioritize it. And then the last sort of metric that we used to decide what got included in the list was which pests might have the greatest impact by either monitoring or implementing specific management actions. So if growers had a little bit more information about one of those two things, what would have the biggest impact? So I'm going to share a couple of examples from this list. So first, I want to talk about two-spotted spider mites. So two-spotted spider mites, it's a mite species that feeds directly on plants, and it can cause damage to a broad range of crops, actually, but that includes strawberries and raspberries. And they're sort of a desiccant in the way that they feed on plants, and it can reduce overall vigor and really result in serious yield loss in really high infestations. And so some of the most important decision-making that happens with this pest has to do with tracking change over time. And the only way that you can really track that change over time is through monitoring. So we decided this was a high impact pest through improved knowledge around monitoring protocols. And here's just a couple of photos of damage, really high damage examples. So we've got a photo of really high spider mite damage in raspberries on the left and then in strawberries on the right. The next example I want to share is yellow rust, which is a pest in raspberries, and this is a fungal disease. It can infect any growing part of a raspberry plant, although it mainly targets the leaves and can result in reduced plant vigor and yield. And the decision-making dealing with this particular pest really has to do with identifying the most ideal life stage to implement management. So sprays, for example, are based on the timing of spore release. So the photo on the right here, you can see that those yellow spots are a little bit dusty in appearance, and that's an indication of sporulation. And so if you're going to choose to spray, you think that things have gotten to a level that they're of high enough concern, you want to make sure that you're implementing that action at the right time. Also, other ongoing preventative non-spray management options for this particular pest can be quite effective if they're timed well. So that's another one that we chose as a high impact possibility. Okay, so let's talk about 2020, this past season. This is when we were doing test runs of the field guides with growers. And I have a few sort of main pieces of feedback that we got from growers that we tried to take into account and make some edits to our field guides and the way we were approaching them. So the first kind of takeaway that we had was around the importance of decision making. So what growers were really looking for was what are the risk factors so that they know what they need to prioritize most. And this was really, the reason I include sprays in brackets here is because for a lot of berry pests, having decision-making leading up to, do I need to make a spray, was pretty important for a lot of the growers that we spoke with, either because they were trying to minimize their sprays as much as possible from a cost point of view, or just from a sort of like a management ideology point of view. So that was a big thing that we incorporated a lot more, was these risk factors. We also learned that a lot of small scale berry growers do use the BC production guide. Also we learned from a number of the growers that we worked with that they do seek information from their chem reps. And so it's important to think about this for us when we're creating resources to make sure that they actually get to the grower. So we're considering, is there a way that we can collaborate with the production guide essentially to post this information and make it accessible to growers. Oh, yes, also, and crop debris management came up as a big question for a number of growers over and over again. So basically what to do with infected crop material, mostly related to diseases in the season. Is it something that, you know, as a grower, I can choose to leave until the fall and deal with when I have more time? Is it high priority and I must deal with it now? Can I till it in? Is it safe for me to add to my compost pile? How do I prioritize management of this? So we tried to incorporate that a little bit more as well. Okay, and then a few other comments I want to include that were sort of outside of the scope of this project, but I think that there are some folks in the audience who are interested in similar work, and so maybe this could be some points of inspiration for you. So one comment that came up from a grower was around demonstration videos for certain protocols like soil sampling or nematode sampling. Yeah, just easier to accomplish how to describe something through a demonstrative video than in a written form. So that was a really interesting thing to hear. Some comments about how it would be really useful to have a field guide really similar to these IPM guides that we've created, but more around irrigation management in particular. That was of interest for a number of berry growers. Oh yes, and then we just heard over and over again just how valuable the in-person service was that that came as a part of this project by us being directly on farm and being able to help identify issues and speak to the growers directly. And I think this maybe speaks to a greater need for some of that extension and outreach for a lot of these growers. So those were some comments that came up a number of times. And I just want to also speak to a couple of assumptions that we had going into this project that proved to not quite be as true as we perhaps thought. So, you know, going into the project, one of the things we realized that all growers would be different, but we sort of assumed that the scale, being small scale, would make small scale berry growers and small scale veg growers more similar to each other in terms of some of their needs and their approaches and the challenges that they were facing around pest management. But what we found to be more true actually was that some of the smaller scale berry growers had quite a bit more in common with the larger scale berry growers than we had anticipated in terms of challenges that seemed to be true for them or ways that they were accessing information and that sort of thing. So that was really, yeah, a useful assumption for us to have kicked. And then another assumption that we had, so, okay, one thing that we heard from a lot of growers basically was that they really valued clarity and non-biased information, which is unsurprising, sort of something that we already know, but it just was emphasized to be even more important than I think we had assumed. It just, yeah, was such a clear and important priority and need for these growers. And I had one conversation in particular that really stood out with me with a grower where I was we were talking about translation of materials and how, you know, having things be accessible via language being really important. And I was coming in with lots of big assumptions about how that was the most important thing. This grower, his first language was Punjabi and was an English language learner. And he was like, yeah, those things are important. Of course they're important. but it was clear that I was placing so much more emphasis on that than for him. What he really valued was having third-party information that didn't seem to come with some basically like ulterior motives for why that information might be provided. So that was a really meaningful conversation for me. Okay, so 2021. Field guide release. We're hoping to put these into the world so they can do the job that they were designed to do. This photo is obviously not from this last year. I find it jarring sometimes to see photos of people so close together these days. So field guides, we're hoping to distribute them as far and wide as possible via farmers institutes or growers associations, etc. And I want to make a plug here that if you are interested in receiving these field guides, please contact Marjo. I've just put her email up on the screen and I'll show it again later too. One thing that's particular about this project is that we have a whole year for the release of these field guides, which is different from the typical timeline for other projects where you kind of have one point of release for outreach. So we want to try to take advantage of that as much as possible. The other part of releasing these field guides is that the project has IPM workshops as a part of their deliverables. And originally we had 2 workshops planned, one in the Sea to Sky and one in the Fraser Valley. Of course, in the face of COVID, we kind of need to re-strategize this, and we might actually be using that demonstration video suggestion that we got from a couple of growers in this project to deliver that in a different way. So stay tuned for that. If you're interested in those IPM workshops, whatever form they take, again, please get in touch with Marjo. Her email is there, and I will show it again in a moment. So in summary, if you fell asleep through all of this presentation and you're just tuning back in now, the 2 things that I hope you take away from this is that the direct consultation that we were able to have with growers in their fields was invaluable, I think, to our ability to make these field guides good and accessible. And get excited about the field guides. They are on their way. And I want to thank the many funders of this project, the BC Strawberry Growers Association, the BC Blueberry Council, the Raspberry Industry Development Council, the Lower Mainland Horticulture Improvement Association, and CAP through the Climate Action Initiative. And a huge thank you to all of our farm collaborators. Yeah, these field guides truly would not be what they are without you. So I'd like to take some time for questions, and I have some questions here for those of you in the audience. If you're needing your brain to be jogged a bit to think about what you might want Marjo or myself to talk about. Also, if you just have comments or discussion points, they don't necessarily need to be questions. I think there's some folks in the audience who have sort of some expertise and experience in this realm. So we'd like to hear from you too.
Wonderful. Thanks so much, Dru. And as you mentioned, Marjo is joining us for the Q&A portion here. I do have a few questions that sort of trickled in as we went. Would you like to start with those, or would you like to start with your own?
No, let's start. If there's questions out there, let's start with those.
All right. Well, maybe a good one to start is you kind of mentioned you focused on the Lower Mainland and Fraser Valley. And so there was a question if these guides would also apply to berries and small fruit grown in the Okanagan and Similkameen.
Yeah, so I would say that they would apply. The one thing is that perhaps the pests that we chose might not be the top pests that are of concern in those regions necessarily. So that might be one thing that feels like an omission. Also, yeah, we've tried to comment on like environmental impacts, like temperature, humidity, and how those might impact some of the pests in general terms, and not so it's just talking about perhaps the more humid region. And that was actually one thing when we were conceptualizing this project that, you know, Marjo really prioritized was having these multiple regions, because the Pemberton Valley and the Fraser Valley are pretty different. And so that would sort of challenge us to not just get too focused in on the needs of one particular area and talking about it in really specific terms. I don't know if you have anything you want to add, Marjo.
Yeah, no, I think you answered that well. Yeah, Pemberton is much drier and also hotter. It's quite hot in the summer, so that's how it's different from the Fraser Valley and Endo. So I think these guides will be applicable throughout BC, depending if you're dealing with that specific pest or not. Yeah.
Awesome, thank you. And then we had a question that you mentioned the decision-making tool kind of is for the pesticide application. And the question is, the questioner is wondering if the scope of the work would also allow for decision-making thresholds for biological controls.
Yes, absolutely. I'm gonna let Marjo actually answer this, if that's okay, Marjo.
Yes, absolutely. So we used, as Dru mentioned, I think earlier, this is not bringing in any novel techniques, but we definitely used whatever threshold was available in the literature. So it's based on what's there. In terms of beneficial insects and maybe release, we've definitely included this aspect. An example is with two-spotted spider mites. Definitely we, included in the guides that predators can be released, and that can be an effective tool. But yes, we use what was available in the literature.
Great. Questions are trickling in. It's awesome to see. We'll see how many more we can answer here before we move on. Have you considered, or would you speak to the need for climate adaptation to pests that are expected to come to BC? For example, as the climate shifts, new pests may migrate from one region to another.
Absolutely. Yeah, not to harp on the spider mites, but I'm going to use it as another example. So spider mites are a great example of a pest that really thrives under hot and dry conditions. And so if as we increasingly see those conditions in the summertime, they're certainly anticipated to be an increasing challenge. And so that's just one example of one of the pests that were included in this project that would be true for.
Yeah, and I think mummyberry is another example in terms of Dru mentioned that we focused on pests where monitoring can really make a difference. And I think mummyberry is also important where your typical calendar spray might not work in a changing climate. With mummyberry, the crop stage is very important. And so that might shift. And also, are the mummyberries in the ground releasing spores? And it's affected by the climate and the season that we're having. So I think those guides are very much sort of designed to address some of the pests that might be becoming more and more of an issue, or where you really need to take into consideration the climate that you're having and conditions that you're having that year.
And I think that's sort of like a reflection of the grower's desire to have the risk factors clarified or highlighted in different ways so that they're sort of equipped to make those decisions in the face of change and in the face of uncertainty, rather than this is just what you should do, here's the answer and do it. But what are the factors to consider and how do they align on your operation, in your season, in your region? So I think that the growers kind of see that for themselves as this is the information that we need to be able to respond effectively.
Great, thanks. And there is a couple more questions, but in the interest of time, we will be moving on. Dru and Marjo, you are welcome to go into the Q&A box and provide some answers in there. And yeah, please keep the questions coming in the Q&A box. And I see there's also a vibrant chat going on, so that's great to see. So thanks again, Dru and Marjo, for joining us.
Thanks, everyone.
We'll switch over to our next presenter. And we're happy to have Dr. Rishi Burlakoti from the Agriculture and Agri-Food Canada Agassiz Research and Development Center join us. And Rishi is a research scientist in plant pathology. His research program focuses on the new and existing diseases on high-value horticultural crops, including berries, field and greenhouse vegetables, all in BC. And his presentation today will focus on decision support tools for managing diseases in berry crops. So thank you for joining us, Rishi. The floor is all yours.
Thank you so much, Nadia, for a good introduction to me. So today, I have two parts in my presentation. In the first part, I'll talk about some unique challenges facing with the modern agriculture in terms of the plant health, and also talk about why decision support tool will help to manage the disease. And I also provide few examples of some of the decision support tool. Some of them are widely adopted in some state of US, including Florida, especially for strawberry fruit management. And in the second part, I will just provide overview of my research program, which focus on this objective, especially evaluating the decision support tool, disease forecasting tool. We are targeting for Botrytis fruit rot in raspberry and blueberry. I'll be able to present some of the field results from this year. Although we have restricted year this year, but I thanks to ARC management, they gave us a little bit opportunity to continue our field research. Finally, I want to just discuss about the future research and collaboration we need if we need to deploy this kind of disease forecasting tool. So just giving you a little bit overview for the broad context, like what are the challenges for modern agriculture related to plant health. So, we have genetically uniform plant over larger areas. That means we are having less diversity in our crop and also cultivar. Another aspect very important for us is when a breeder try to release the cultivar for the better yield. And it's a universal fact that when you try to get the trait for the high yield, then you will lose the disease resistance automatically. And that makes more challenge. Another one, in the developed country, we have continuous monoculture or few related crop. And if you have, especially like in the berry crop, you don't have option. But even on the vegetable, if you have same, crop over and over, then you will have big problem with some of the soil-borne disease. Another aspect, if you go for the management side, we are using more synthetic fertilizer, pesticide, which will deteriorate our diversity. And overall impact, disease severity or susceptibility is increasing. And we are also having some pesticide resistance problem with the pesticide. And definitely we have environmental impact on this. So being said that, we should have come with some resilient and sustainable disease management approach and tools. So this decision support tool or disease forecasting tool might be one of the options for our sustainable disease management. So why disease forecasting? Definitely it will provide advisory to growers for appropriate timing of the fungicide. And if weather condition is not favorable, and then it advised like not to spray, means it will decrease the number of sprays. And also it might help a little bit to reducing the fungicide resistance problem. And definitely cost of production will be reduced if there are good disease forecasting system. However, it has limitation too. So if model is not robust and not validated well in the regional level, it might give you false result. So Briefly, I want to talk about what kind of factor we can consider while developing or deploying this decision support tool. So this is a little bit elaboration of the disease triangle curve. In the disease triangle, there are basically host, pathogen, and environment. However, if you talk about a little bit broader context, there are human activities, and over time, it's kind of confounded. So for the disease forecasting, always any moisture, like either that is form of rainfall, high humidity, dew, irrigation, fog. This is the number one factor for the disease infection and more disease pressure. Of course, temperature is also key, play the key role. But however, when you have combined moisture and temperature, and it has a broad effect. Definitely, the environment is one factor, but you have also host side. If you have more susceptible and genetically uniform crop, then there is more risk. And another side, in the pathogen side, sometimes environment might be conducive, but if you don't have enough inoculum pressure or type of the strain, if some strains you have more virulent than the less virulent, it will impact. So anyway, any good model, if we can consider and incorporate all these factors, that will make a more robust model. So now I'm going to provide you few examples. Some of them, as I told, is widely deployed at the larger scale. So one is that in Florida, they started in the beginning just deploying this forecasting model, especially targeting Botrytis and Anthracnose fruit rot. So, Florida, they have very high humidity and they have all season. That's why they have really big disease pressure with this fruit rot. So they did many research, as I told, it started from Florida, and then they moved back to the several eastern states. In this next slide, like now, after combining all these studies, they have this decision support tool, which is available in the mobile version and also in the web version. So this is how the that decision support tool display in their web version. So if you see one of that dot or that map, and you have option to click there, and then grower can see what level of the disease risk during that day. And then they also provide some recommendation what to spray for that to control that rot. So now I want to just talk about my previous research work. We also validated that Anthracnose fruit rot forecasting system. This is the same model Florida used, and Ontario. And if you compare the weather, Ontario versus the Florida is more humid and it's quite similar. So basically, our objective was to evaluate the weather-based fungicide spray program with the calendar spray to control the Anthracnose in the day neutral strawberry. So I want to go quickly about the result. We conducted research 3 years in the field, and then we compare model-based spray with the calendar 7 or 10 days spray. And basically, we found the model-based spray was quite effective as the calendar spray. However, in 2010, there was variability in the result. That is due to fungicide itself, because the captain we use in the model was not very effective. That's why in 2011, we switched with a different product in all treatment. So based on this result, then we recommended to use this decision support tool, which worked well for the Ontario strawberry growers, and it was deployed for the grower. So there is another approach for the decision support tool, and people, especially in Europe and many parts, people also use mechanistic model. For me, it is also complicated, and you can see they use a different information about the life cycle of the pathogen and tally with the different growth stage, infection cycle, all this fancy tool, and they come with that. But for me, when you have more input variable involved and more complex system, then you might increase your chances of error in the all aspects. So anyway, my approach I'm not using right now with this mechanistic approach, but just give you information. This is one of the approach. So now I want to move regarding our effort, what we are doing here to evaluate this decision support tool. So as I told, the major focus for the botrytis, and everyone knows botrytis is a problem for raspberry and blueberry, and it not only causing the fruit rot, but also cause cane botrytis with this pathogen. Dru already talked about this mummyberry, that is another problem, and we have also anthracnose. However, in this presentation, I'll focus more on the botrytis side. So, as I mentioned, this is the one of the objective of my berry cap project, supported with that Agriculture, Agri-Food Canada, and berry industry. And we use two approaches. One is conducting the field trial in research station, primarily focusing in Agassiz and Clarebrook, and also in collaboration with the farmer. And thank you so much for ES Cropconsult guys. You are always phenomenal for me to provide information and sample all this. So anyway, in farmers field side, we have also identified in the different area the farm, and also they have the weather station we need. However, one challenge for grower field is we have hard time to find the on-sprayed part in the field. That's why we are also trying to focus a small farm and organic farm where they use less spray. Anyway, in 2019, we started to compare some of the model in the website internally to see how this different model will work. And in 2020, we started to conduct the field experiment to just compare model-based spray program with the calendar program. So I want to just highlight here, I don't want to spend much time here, but I'm also involved in the part of this biovigilance project led by one of our scientists from Quebec. And in this one component is we are also using that spore trap where we can use qPCR technique to quantify the pathogen. So I'm also aiming to include that spore trap data, spore monitoring data we can use in the disease and weather data to aid in this decision support tool. Unfortunately, in 2020, this project, we didn't get chance to work. However, I tested that equipment in the field and it's running pretty well, but we were not able to analyze any data. Just sitting in the -80. Our collaborator from Ottawa group, they are helping us to do this genomic work on this trap. I'm hoping next year we can speed up a little bit if we don't have much restriction. So in this part, I will briefly explain. It's little, I apologize that there are a lot of graph and a lot of information which might be boring for you. However, I will try to simplify that. However, what I want to clarify this one is I want to talk in this part based on the knowledge on the literature. I also created some model myself and also use the previously validated model, and using our weather data from Clarebrook and Agassiz, I just try to run how does model perform. Okay, and then the second part I will come what we did in the field trial. So I use 5 prediction model. So just keep in mind, these models were developed for strawberry and grape. And for my knowledge, there is not a robust model for blueberry and raspberry. However, I created 2 models which calculate daily disease severity value using the temperature and leaf wetness hour using the published paper. And I use that 2 models. and also use previously widely validated model. So as I explained here, the model one, based on the knowledge, I created that daily DSP value and then run. And I use that model for our field trial to make a spray decision. So model two, as I told, I just explained about that Florida model. So it is widely validated and deployed in the Florida and eastern part of the US. So model three, this is for Anthracnose. Again, it is widely deployed in the Florida and eastern part. And the model four is, I just created that DSP value based on the Jamir Punja's lab. They did some epidemiological study on Anthracnose in blueberry. And the fifth one is the model for Botrytis on grape. So this graph, the first two graphs in the upper part, you can see this is the daily DSB with the bar, and then the graph is the weekly accumulated DSB. And the lower two graph, this is the daily leaf wetness hour. I want to just compare how 2019 and 2020 performed. So, you know, in 2020, we get a lot of rain and we got also a lot of disease, a very good year to conduct the pathology trial. But 2011, we have comparatively less dry summer, dry summer. So basically you can see here, 2020, the disease pressure, the disease severity value daily and weekly. If you see from here, the 1st June to the until the end of the July, it showed very high DSP compared to 2019. And you can see here a pattern in the leaf wetness hour is also higher in 2020. So this graph, again, comparing the model performance of 3 already available model in 2019 and 2020, I gave you in the down the threshold. Don't confuse here with the only value because threshold is different for different models. For example, broom, that is for the grape botrytis infection. If you have that infection value is from 0 to 1, if you have less than 0.5, then low infection. So mostly we see in both here infection level between 0.5 to 1, and it is higher in the 2020 and low in the 19. So it showed some pattern. Same with the bulgur, that in botrytis model, we have little bit higher in 2020. Surprisingly, Wilson et al. Model, that's for Anthracnose, it didn't perform well in my mind because in 2020, you can see still a very low infection risks, which is not true in our condition. So my thinking here is the pathogen we have can adapt pretty low temperature compared to the other part, which we need to do further research to validate. That's why these models are not performing very well, but my plan is to continue this validation work. So now I want to move in the third part for the field evaluation using model-based and calendar spray program. So we conducted 3 trials, 2 in the raspberry and 1 in the blueberry. But keep in mind, I'm just talking about only 1 raspberry field trial due to time limitation. So basically, the model-based spray program, I already explained that this DSP approach I created based on the knowledge. I use that and also use some information about the rainfall and other weather patterns. So to make a model-based spray treatment, whereas for the calendar spray program, we regularly spray between using that 10 to 12 days interval. So basically, number of sprays were same because we had very wet weather condition. However, you can see some timing different in the calendar versus the model-based program. So just sharing result. I just use 2 simple parameters here to display the result. So one is the fruit rot incidence. We harvested twice a week and then counted the berry and then infected versus healthy, and then we just calculated fruit rot index incidence, and also we talked the marketable fruit yield. So basically you can see both calendar and model-based treatment performed very well over week to reduce the fruit rot incidence, whereas fruit yield was higher and those treatment compared to the control. And if you see the cumulative fruit rot incidence and cumulative fruit yield, you can see the similar pattern here too. So finally, I want to just mention a little bit about our future research. So we want to continue the same work what we did in 2021 and 22. And I didn't present here, but we are also collecting some data from farmers field. But we found a little bit tricky in the farmers field because, as I told, we cannot get the on-spread plot. However, we are focusing small farm and organic field. So in farmers field, we have also weather station, and then our aim is also use farmers field data, both disease and weather data to validate the result, which will be definitely integrated with our on-station research. The final part, I'm expecting the long-term collaboration with the industry, especially berry industry and provincial government, BC Ministry of Agriculture and other collaborators. Let's see if we have model, then how we deploy is a regional level to support the disease management for the grower. So that is the discussion part I want to keep open always. So finally, I want to thank our funding support, Canadian Agriculture Partnership and berry industry. Then I would like to thank all berry growers, our staff, and our project collaborator and industry partner. I don't know how much time I took, but if there is any questions, I would be happy to take it. Thank you.
Thank you, Rishi. That was very insightful. And we do have one question here, and it's related to kind of the microclimate and the fact that it seems that your models are very dependent on accurate weather data. And so Kathleen, who asked the questions, she's mentioning an example in Nova Scotia where she had a case of one farm experiencing flooding and another farm only another mile away experiencing extreme drought in the same season. So the question is there a concern that weather data may not be collected at high enough granularity to be useful for forecasting as climate change leads to more extremes in small areas?
Yeah, that is true. Like always there, especially, yeah, like same with the Nova Scotia, we have also a lot of microclimatic variation. That's why I didn't explain well about the weather equipment we are using. So that's why what I'm doing in this research is, let's say in Agassiz, when we have the field trial, we have the all weather station with the all sensor here. And I didn't compare the result due to lack of time. We have also in the clear book, we have the on-site weather station, and we are also collecting some disease data from there. And also I talk about different location in the farmers field where they have the on-site weather station, and I added in that one leaf wetness sensor too. So what we are doing in this research is from where we got this data, we are considering those microclimate variations. So using the on-site weather data, especially in our research trial in our on-station, and also in the farmers field, we are trying to use at least like few kilometers, like maybe 5 kilometers within that area using the weather data when we have the disease data. But I agree about that. We need to consider that microclimatic variation.
Yeah, definitely an ongoing question as we work on various microclimates. Oh, Dru has a question.
Question for you, Rishi. I know that you didn't end up using, or it sounded like you didn't end up using the spore trap for Botrytis, but I'm curious if you could just explain that, how that would work a little bit more. And part of why I'm wondering is because we've been doing a project in spore trapping for late blight in potatoes, and that is really based on like just presence of those spores, of any of those spores is of high concern, but Botrytis is fairly widespread, pretty ubiquitous. And so I imagine that you'd be looking for spores in a really different way. Could you just explain that a little bit more?
Yeah, thank you, Drew. So traditional way of spore monitoring is we use the spore trap and then you count the number of spore in the microscope and quantify how much spore you are getting daily, hourly, weekly, whatever way you want. So there are two kind of basically, like the spore trap. One Burkhardt, where traditional way you use the tape and then you have timer and rotate hourly, or you can take data hourly or weekly or daily, whatever. Another one is just that rotor sample, where you cannot get hourly, but you can run for certain hour in one day and change that and use that and count the spore. But my approach here is it's a lot of time consuming to do the microscopically counting spore. So we are trying to use qPCR approach, where we can use that spore trap and do the qPCR and quantify how much spore we are getting. So I'm planning to use both Burkhardt type, which is quite expensive, and in the Burkhardt, when I showed it picture, you can directly collect the spore in the tube. And that tube, use that tube for the DNA, like extraction and use for qPCR. But also people use that rotorod sample, and we already have method, especially scientists from Quebec, she work on that in Raspberry, and she used the rotorod. That's quite cheaper than the Burkhardt. So anyway, my plan is compare both method and develop that, use that qPCR. However, with this restriction, I don't know how much we can do next year, but I'm hoping. I already have equipment and we are working on the protocol, validating protocol. So anyway, that is the approach you can use to quantify.